Okay, looks like we're going. Do a quick audio check. Audio check, audio check. Okay, looks like we're going. Do fire up a check. Some music from my friend JX3D, also known as Henrik. Used him before on some streams, and I wanted to try it again because I think it worked out well. Okay, looks like we're going. Do fire up a check. Some music from my friend JX3D, also known as Henrik. Used him before on some. Wanted to try it again because I think it worked out well. Okay, looks like we're going. Do fire up a check. Oh, I see what happened. I'm getting a loop back on the audio here. JX3D, also known as Henrik. Used him before on some. Wanted to try it again because I think it worked out well. I see what happened here. That's not playing. That's playing. I think it's just because I'm listening to the repeat. Let me just do a quick check on my phone. Make sure everything is good. Let me. Used him before on some. Wanted to try it again because I think it worked out well. I see what happened here. That's not playing. That's playing. I think it's just because I'm listening to the repeat. Let me just do a quick check on my phone. Make sure everything is good. Yeah, I'm getting a loop back because I'm actually monitoring it on the stream itself. So let me kill that. And my repeat text, my repeat chat should stop in about a minute. Okay, now it's not repeating the text. So here comes the music from JX3D. This is from his album. Should I get this right? Because it would be bad if I got it wrong. Whoa. Why is it not? Where did my folder go that's playing it? Hold on. Oh, it's in the browser. That's a different browser, that's why. Hold on, bear with me. I'll figure it out here. Just give me a second. I'm playing it from his SoundCloud. I mean, from his stream, not StreamYard. His Bandcamp site. That's it. Brain is now engaged. Bandcamp site. And it's his album. Wait, is that the right one? Hold on. I'm all kinds of turning around here. No, that's not it. Worlds Apart. Sorry about that. GX3D, Worlds Apart. Bandcamp.com, GX3D. You can look it up. And here we go. Get the balance on it, right? Turn it down just a tad so you can still hear me talk. Okay, I think that's a good level. You guys in the chat, I think the balance is off. If you're here, let me know. I'll be checking the chat shortly here. Okay, enough fiddling with all the settings. Let's get to the nitty-gritty here. So, started with some little botany this time. I've done some plant stuff for a little while. With a lot of fall color stuff in the area lately. We don't normally have fall color because a lot of the plants here don't. The temperature doesn't change as dramatically as it does on the East Coast and some other areas where it gets from hot to wet to snowy to icy and all that stuff. And so for lots of reasons, to the temperature and just the plants that pretty much evolved in this area just generally don't do that as much. They generally do, or if they do, they go straight from a green to a brownish color with very little in between. There's basically several reasons for that. I'll go over that real quick. So this is the green leaf of the Pistachia sinensis, the Chinese pistache. It's a relative of the pistachio. That's not really edible to humans, but birds eat the fruits and stuff. But we don't because it's not really cultivated, and it's ornamental. It's very pretty this time of year as it's going from red to yellow to orange and green, orange and reds, and then the leaves start to drop off because it's a deciduous tree. And there's some good reasons for that. Let me go over that real quick here, just so I don't miss some of the details. I wrote up a quick little thing explaining the process. So as the days get shorter and the cooler temperatures come in, a bunch of the biochemistry of the plant changes. So essentially, those two things signal a release of a hormone in the plant called abscisic acid or abscisic acid, and it makes the cells at the stem of the leaf connection to the plant. So if it's a cluster of leaves and not a single leaf, or even just a single leaf, depending on where that abscission zone is supposed to form, it's different for each species. It has compound leaves or simple leaves. It forms an abscission layer. It's kind of a little corky layer of cells that essentially sounds like what it does. It kind of blocks off the leaf from the rest of the plant. And so when this layer is formed, you're still doing photosynthesis from the green pigment, from the chlorophyll, and the different types of chlorophylls as well. But in general, from the chlorophyll, which makes the plant, the leaves green, and in some cases the stem green. Some plants have green stems as well as green leaves, so they do photosynthesis on the stems and the bark itself, on the trunk. So the abscission layer basically makes sugars accumulate. They can build up in the leaf, but they no longer get out. And so a combination of the sugars accumulating and the leaves, getting ready to detach based on the abscission zone, basically the plant, it stops making chlorophyll. And it's also, when you study this stuff, everything's about energy balance and how much, like, for something that does a reproductive cycle, it's wasteful to do too much of one thing when you don't need to. So, for example, a natural fruit like a cherry, if you get a natural cherry, not a cultivated cherry, and it's a huge pit and a very small amount of fruit on the outside. It's a waste of the plant's energy that's all about reproduction with the fruit to put more fruit than it has to around it than the seed, because the seed is the reproductive part. You want to continue your species. And you give just enough reward to the bird or other animal that will pass it on for you to plant it and have your next generation. We cultivate it because we want yummy fruits. So we make the pit smaller and the fruit part bigger. So we make the plant expend a lot more energy for something it doesn't need to do. It's just something we've made it happen. So similar with the photosynthesis, if the leaf is no longer going to be useful, it stops making photosynthesis because you build up too much sugar. The abscission zone is already formed. It's a wasteful thing to make more chlorophyll. So essentially, chlorophyll production stops. Now, in a plant that has lots of other pigments underneath, yellows, reds, blues, purples, oranges, those get revealed as the chlorophyll breaks down and stops getting produced. But in some cases, like there's an oak tree, I believe it's called the, where did I put that down here just to make it clearer? There's an oak tree called a bur oak that, like a lot of other oaks, it doesn't go through a bunch of color changes because it doesn't have a bunch of those other pigments in those leaves. So it basically goes from a green leaf to a brownish leaf. And this is before it's dried. It's actually turned brown because of the tannins and stuff in there that have a natural brownish, reddish color. So some plants go straight from green to brown and then eventually drop off and then dry up and die. But other plants go through this flash of colors that everyone's famous, for seeing the fall colors. And so what happens is when the green has stopped production, you get other supplementary pigments used in photosynthesis and other things. And one is called xanthophylls. Those are the yellow pigments. And they're similar to pigments in that makes the marigold yellow and other kinds of yellow pigments in other flowers and other plant parts. So it's actually has a photosynthetic step, but it's biochemistry. I don't want to get into that crazy stuff right now. Oranges are formed by carotenes. So everyone knows beta carotene. It's a good supplement for your diet. It's good for you. Or they're called carotenoids. And carrot's a famous example. Carrot's orange because of all the carotenoids. So you get the oranges revealed, you get the yellows revealed, and you go through this phase sometimes of like more yellow, more orange, and it often ends in a reddish color in some before they drop. So in the case of Chinese pistache, it goes through all of them. It goes from nice dark green like you see on the slide here. This is a dissecting scope. This is a pretty big leaf. It goes from green chlorophyll to the xanthophylls. And then it goes to the carotenes as it's fading into a reddish color toward the end before the leaves drop. And then the reds and magentas are caused by anthocyanins. So quite often that's like poison oak will do that before it drops its leaves. It'll go through the green and the yellowish, and then it goes to the reddish color when it's nice and oily and nasty. And then the leaves will drop. It's a deciduous plant as well. So those are the anthocyanins. Cyano means bluish, right, in general. So it goes from crimson red to magenta to sometimes a little bit darker, and there's a mixture of reddish and bluish colors in there. So anyway, that's what's happening in the leaves. And so now we've gone over that real quick without too much detail. You can get a nice look at these, the leaf detail here. It's almost like a relief map. This is the bottom surface. Let's go to look at at some of the veins here. So, let's see if I can find some other structures on here that are interesting. Mostly just the veins and some of the details. I'm not quite close enough to see the stomata, I don't think. Stomata are, or stomates as people call them, are the mouth-like openings of cells that kind of expand and contract to allow gas exchange. So CO2, oxygen, water vapor, all that kind of stuff. Your plant is constantly, you know, under, like right now I'm shooting bright light at it, so it's gone through photosynthesis for sure. In the process of photosynthesis, you're turning light energy using CO2 and water to make sugars, carbohydrates. Hence the name carbon and hydrogen. And hydrate is hydrogen and oxygen combination. So carbohydrate literally means carbon, hydrogen, oxygen. So, you know, whether you're talking about sucrose or glucose or whatever, those are all carbohydrates and different levels of carbon, hydrogen, oxygen. And so there's also water involved. And do you have gas exchange? So during the day, plants generally take in CO2 and release more oxygen than they use, because photosynthesis is using a lot of CO2. And at night, they go through a normal respiration phase where there's no more light for chlorophyll. So they do what we do. They respire. They bring in O2 and they release CO2. This is why it's often said not just to make your workspace or your home more beautiful with plants around, because there's life and it just looks nice. But during the day, they're also releasing lots of oxygen in your environment. So that's actually good for you. And all due to photosynthesis. So those stomata actually open and close. There's like a lot of cell functions in plants use water pressure. It's called turgor. And you actually get cells kind of inflating, deflating with water. Similar to how, on a much larger scale, how a Venus flytrap kind of closes. It's partly expanding the cells and partly growing at the same time, but it's a quick release of water pressure to make the thing close. So turgor. Anyway, so I'm not close enough to really see stomata here very well. They are there, but I think I've gone as close as I can with this. I have a reduction lens on here. There's a negative Barlow essentially, so I can get further away from bigger specimens. But because of that, my zoom is also limited. So if I'm going to zoom back even further, you can see the whole leaf pretty much, and the tray here. So the exposure's off a tad here. Adjust the light for the exposure here. So anyway, you can see, and I made it so high in this camera I actually can't go that far back and focus on it. So I've got to kind of narrow my range with some custom settings on this scope. I'd have to go in and doctor it some more. So anyway, you get the idea of the texture of the leaves and the color, and this one's actually starting to change. If I go further down here, you see this kind of pigment change here. This one's going from green to red. Turn the color back up here with the lights. So that one's already this is kind of a hybrid leaf. It's going through changes in color bit by bit. It's not like overnight when they change, get these transition zones. So that's the green. Now let's go to the one that has the xanthophylls. Let's pick one of the yellowy ones. And in this case, this is a single stem here. It's actually a compound leaf, so it has leaflets on a stem. They have two general terms. There's lots of other ones, but there's pinnate and palmate. Pinnate looks like a feather, where there's a central spine, and down the spine you have leaves going across. Using these two things to show the cross there. Going across the leaf, and then it ends in a terminal pointy leaf. That's pinnate. Palmate is more like your hand. You've got fingers sticking out in different directions, the leaflets going that way. Those are kind of the generic common forms of compound leaves. But so for example, here's one of the yellow guys. Nice yellow. Get a little light balance there on the color. So we can see the green is all gone, basically. Chlorophyll is no longer being produced. And in doing so, it revealed these pigments underneath. In this case, lots of xanthophylls. Let's zoom in a bit and see what we can see here. Change the light levels a bit. And the focus. So it also shows the detail of the leaf a little better because the veins are darker than the tissue. And when it's green, you don't really see that level of detail. So while it's going through color changes, it's kind of nice because you can actually get a different perspective on the leaf, on the contrast there. So there you go. This one's kind of dirty. Got some injuries on it there. Probably caused by insects. The black could actually be some fungus starting to grow in there because the leaf is dying. And then further down the same leaflet, actually, the same compound leaf, you have a nice bright red, crazy, in your face end of the life. So this one's already starting to degrade too. So the dark spots is probably some fungus starting to come in and break the leaf down as it's getting near its end with the red pigments being prevalent. It's usually they can drop in all different colors, but I found quite often most of the ones that have dropped are the end of the life tends to be when they're more reddish, but it does vary. If you look on the ground, you'll see lots of different color stuff that's not green. And again, nice contrast there with the lines and the tissue. Turn this up again. Yeah, this gets real dark now. It's a cool texture, but it's some of the details pop, but it's really hard. And I can also drop some backlight behind it. Check this out. That brightens it up a little bit. So I have light coming from above and light coming from below. Just like when you shine light through your fingers, you can actually have light transmit through your skin. You get that kind of a glow. So yeah, that kind of helps show the little clusters there, the little bumpy areas quite well. And again, looks like an aerial map of, of a red planet or something. Excuse me. So that's the different phases of Chinese pistache as it goes through fall color. So people are out taking pictures in droves lately because, again, there are only a handful of things that change color like this. Like the local maples will go through some colors. A few of the oaks can change color a little bit, but generally a lot of the plants go straight from green to kind of a brownish color to reveal tannins and other pigments that are more kind of a brownish color. So, whereas, like I said, go back east or some other countries with the weather is quite significantly different than somewhere like California, and you see quite a bit of that. So what I wanted to do after, well, maybe I'll try it right now. Let's see what happens if I grab one of these little light yellow ones and throw it on the other scope, the compound scope, and just flood it with light and see what it looks like. Should be able to see this stomata pretty well. Let me quickly sandwich them between a couple slides because it's curly enough that a cover slip won't work, so I'll have to put 2 slides together just to make it nice and flat and minimize the focal plane. This is what you call a dry mount. I'm not putting any water on it. It's just a leaf smashed by 2 slides. And let me go get this lined up here, and I'll switch the camera over. That's pretty cool with this magnification. So let's swap Peruni here. Okay, let's adjust this here. So this is pretty low power. Similar to the power you already saw on the other scope, and now we're just lighting from below. Transmission microscope. And let me just adjust this real quick because I have a weird offset with what I'm seeing here. I want my camera showing in the eyepiece. I need to be able to have them in sync. Just fix this real quick here. That's good. That's pretty much in sync. Occasionally I have to look for details in the scope's eyepieces in general because I'm seeing a little more detail. Actually, this camera's doing a really good job. This is a Nikon mirrorless camera. I upgraded from my other USB camera that was decent, but not as high pixel count as this or color handling capability. Alright, so let's see if I can adjust everything here a bit. So I'm not seeing anything significant here. So let's go up another level. This looks like some pigment started to form here. See some red starting to form in this zone, but it's still predominantly yellow. So it looks like almost like I've stained it. Well, I haven't done anything but just pick it. Just adjust everything here a little bit. Oh yeah. Let's go look around a tad more here. So you can see the stem is starting to get reddish. All these little bits of debris here. I think it's like insect sheds and stuff. Some really weird little shapes in here. Okay. See what else we can find here. Focus is odd because, you know, it's not completely flat. The weight of the slide is not completely flattening out the leaf, so I'm having to focus constantly as I move around. But look at that little geometric pattern. It's really interesting. Where the regions are broken up there. So the veins are going through here, but you can see the individual cells. I can actually see individual nuclei here. Let me play with this contrast a bit more here. Hey Rob, how's it going? Yeah, got some good stuff coming. I just started with a little botany first, just because I had these leaves from the fall color activity lately. So I'm just going to see if I can push this one more bit. I don't know if I can with the problem is with the slide making it thicker, it's harder to use some of these longer lenses without hitting the glass. Let's see if this actually works. It probably won't. It's kind of there, but it's very dark. So I can barely focus it before we break the glass. It's not quite doing it. Let's try. Pushes it up, so let's go back down. Put that into play and slowly bring it in. To where it just gets in focus but it doesn't break the slide. Let's see where that level is. Should be just about there. I don't want to push it too far. It's not quite doing it. Why is it not doing it? It's like right on top of it. This is the tricky thing with these tiny micro distances, is just the extra thickness of a slide can make all the difference with a higher magnification. So let's not do that. I'll try it with one more leaf. Maybe we'll get some different results. They're all about as flat as they're going to be, so I can't make this too much better. Let's check one that's a little more orangey. This is kind of a transition leaf here. A little bit more orangey. Oh, come on, get in there. Get in there. Okay. See what we can do here. So it's kind of more orangey over here. You can see it's really just a mixture of the red and the yellow making the orange. So let's see if I can play with these contrasty bits. The leaf is still fairly thick, so it's not really transmitting the way I want it to through the camera. You can kind of see it there. I can do a digital zoom here on the camera. It'll help a little bit. I'm gonna find that area. It's a little clearer here. There's all kinds of debris on this one. Might have better luck on I think the other leaf is better. What I was trying to show is hard because the leaf is still fairly thick, even though light is transmitting through it. The cells are stacked on top of each other, so I'm seeing individual nuclei in cells in these little islands between all the little veins. But it's really hard to pick them out as they're overlapping each other. So it was worth a shot, but it didn't work too well. So that's pretty much it for the leaves. We'll look real quickly on the other scope at a couple other leaves I got there interesting, just for laughs. There's a very primitive leaf. Let's switch cameras again. Turn the backlight off. And this should show some nice venation on the leaf there. If you notice, it's parallel. It's like, what kind of leaf would do that? It's unusual. So that's because it is come on, where's my focus? Yeah, this Barlow is making things interesting. This is a ginkgo biloba leaf, also called the maidenhair tree because it looks kind of like a maidenhair fern in a larger leaf. So it comes with a narrow stem here, and then these veins fan out. And you get this kind of a scalloped end here, and these diverging lines basically forming the leaf. It's a very distinct leaf. So this one's kind of it's weird. It's kind of yellow, but it's showing more green on this for some reason. Let me just zoom out a little bit. There you go, it's better. So that's the typical ginkgo leaf people are familiar with. Turn it over. See, it's already starting to die, parts of it, because it had changed color. This yellow color before it's dropping. So it's going through the xanthophyll stage. It's normally a nice green leaf, and it's going through the yellow as well. So interesting texture there. Just taking some quick snapshots to see if that actually works during the stream. Okay. So that's ginkgo biloba. Nothing special to report there. You guys probably know about they use it medicinally now to kind of essentially open up your capillaries in your brain. It's supposed to get better blood flow to your brain. It's supposed to make you think clearer and more, all kinds of the typical, catch-all that they do when they talk about some of these medications. It's kind of funny, but like a lot of these things, it's also not good for you at certain concentrations. It is a toxin, so you want to be careful with that stuff. This is interesting. This is just a little plant in the area called Toyon that it's not the best specimen of it, I have to say, but it's got some nice little fruits on there, and has this kind of a serrated leaf. I showed on some of my hikes. You said the live streams where I did hiking at Edgewood Park and stuff like that, but I never put it under the scope before. So it has these little red berries that the birds seem to like. I don't think even Native Americans had a use for this, maybe for a dye, but I don't think it was a food source. I'd have to look it up again. Used to know all that stuff, but I forgot. But yeah, there's nice little green leaves, kind of leathery, and these red fruits. Very distinctive when you go walking in the Chaparral Oak Woodland area around here, the foothills and stuff. Let's turn the leaf over. That's the top of the leaf. There's some more light here. So heavy, shiny, leathery coating on the top because it's in hot, dry climates. Lots of sun all day long. It's got to have ways to store water as much as it can. There is transpiration, as I explained before, part of photosynthesis and just normal respiration, just like we breathe out water when we respire, and it's vapor, and they also breathe out vapor, which evaporates up into the air and basically collects eventually into clouds, just like all the other evaporated water from waterways, rivers, lakes, streams, stuff like that in the ocean. So transpiration is a big part of it, and that's why by deforesting an area, not only do you remove the food source for animals and the habitat for animals, but you also remove the oxygen developing region of a landmass, and also more microclimate stuff with the moisture being released and overall humidity of the zone, and just being habitable, being nice, being pleasant for animals, including humans, to enjoy. So deforestation is a real serious problem. It's not just an eyesore. Most of the oxygen comes from algae, including microscopic algae in the oceans and large waterways of fresh water. But forests provide a huge amount. So just by cutting out huge swaths of rainforest, and whether it's tropical or temperate or like the Pacific Northwest, where they cut out giant clear cuts and stuff, it's bad no matter how you slice it. And so greedy people want to just have housing and lumber and all kinds of multi-use areas, instead of letting a nice forest being there that's been there for thousands and thousands and millions of years and turn it into a big clear-cut ugly zone like a nuclear bomb went off. Is really sad. But it's also sad for all of us because it does affect the microclimate and the overall climate. Like I said, less water is being released into the environment, into clouds, and just microclimates in general. Humidity, overall local humidity, local temperatures are all affected by that, and just habitat and food for animals. So Tolian is a nice little bush that grows around here. I just thought I'd show that real quick because I never put it on the scope. And what else do I have here? Oh, a couple of succulents they had. And I was hanging out with my buddy Marlon at the radio station the other day. With some interesting succulents on by one of the offices there. And I don't remember this specific succulent, but they're pretty common. Long, thick leaves. They're not like jade plant. It's a different one than that, but there's a little tapered one, almost like a little like a green bean kind of looking thing. But it's just I broke it off. It's a piece of the succulent branch, and then this is the other flatter one. I'll turn it over. You can see the other side. It's not that different on the other side, but at this scale you don't see too much, but when you zoom in, start to see the waxy protective layer again. So this has succulents to store water, but it also has and it's thick, so it has a lot of volume to surface area. But it also has this kind of a waxy coating on the outside. You can feel it when you touch it. It's kind of grabs you a little bit, kind of friction. And you can see it here. It's a kind of a waxy coating. All these adaptations to hold water at all costs. And the plant has to produce all this stuff. So this does cost the plant energy, but it's all in the nature of storing water and dry habitats. That's a good thing. You want to do that. That's worth putting your energy into those structures and those chemicals. So now they're starting to see some individual little little spheres there. I don't think that's quite the cell layer though. This is not that high magnification yet. But one of these days I'll get my microtome out and start doing some thin sections of some of these kind of leaves. So you can get into the inside and get a nice look inside the cell. Inside the cells of the stem. Leaves and stuff. So we've got color changes, we've got adaptations to moisture loss. So that's the leaves kind of part of the show here. A little bit of botany, not too much, not too fancy, not too nerdy. But then I went to where I collected some of these leaves. Not the pistache, but these other leaves. And there's a little waterway, a little kind of a pond structure at Foothill College. And I wanted to see what's in the little kind of pond structure they have there. It has a lot of horsetails growing in it. This one had primarily rushes, which is the round tubular guys with little dry flowers on the tips. It had some other water plants, had some water lilies in there, and seemed to have a decent amount of stuff moving around, or as far as giving it color. So I figured there's got to be some good guys swimming in here. That's a perfect place to have it because it's right outside the biology labs at Foothill College. So I imagine if they're clever, they don't just have it as a beautiful thing to look at with some plants and some greenery and stuff, but they also go out and get specimens for their labs. And that's what we're going to do right now. We're going to look at some specimens from the water feature there. See what we can find. Move this over here because it's in the way. Okay, too much light. Alright, now let's find... Whoa. Some stuff moving around already. Probably put too much water on the slide on this one. But we'll see. Okay. Let's see what we can find. Yeah, it's a different... I'm on the wrong focal plane here. Let's go down where the good stuff is. Just a little dark field first, just for fun. Helps to find the little tiny critters that are quite fast at times. Oh, there's something. Very small flagellate bouncing around. Is that a guy? Oh, the frame rate's kind of weird on this. Why is it doing that? Shouldn't be jerky like that. Let me fix that. Interesting frame rate. See if it continues to do that now. Yeah, even my scrolling is a little bit choppy. That's not good. Alright, let's fix this. That should be a little bit better. Let's see if that improves the frame rate a little bit. Yeah, that's better, but it's still... Still a little choppy. What's going on there? I wonder why that is. I didn't notice that on the last stream with this camera. a little better now. Let's see. Let's see if I can zoom in real quickly digitally on this area without having to tweak the lenses. See what this guy looks like. It's definitely a little, you can see the greenish color. It's definitely a small green flagellate, so it's a microscopic plant. Talking all the junk in here that I have to focus through. A whole bunch of these guys here. Look at all of them. All over the place. Ah, there's a frozen one. I don't know if it's resting or dead. Let's see. Let's go a little closer and see what we got. Just sitting there. It's not doing anything. Yeah, my frame rate's a little off still. It's odd. Let's do a digital zoom while we're here. We can study this guy real quick. So you can see the nucleus there, but it's no longer swimming. So it's either expired or taking a break. Let's try one more digital zoom because we can. And you can see the nucleus very well there. Let's take it out of oblique. Now you're getting more of the green. Okay, so it's still got some activity here. See, the cell has some little Brownian motion going on inside of it. So that's what it looks like up close. Now if we can go back out digitally, watch some of the friends swimming. And we can get a close shot of them swimming in a small pattern. Yeah, they're going in and out of focus, up and down, all over the place. Well, at least one was stopped, so I get a nice look at it. These guys are on the move. But what's sad is it's still choppy. And I don't know why it's choppy. I didn't change any settings from last time. This guy's moving slowly. That'll help. So I do a digital zoom on this guy. You can watch him a little bit better. So it has that kind of jerky motion. It's fairly linear, but it's a wobble because it's using at least one large flagellum to swim, and that makes it kind of wobble as it whips it back and forth. That's the immediate way you can tell it's not a ciliate, it's a flagellate. Ciliate's a very smooth, purposeful movement. It can turn on a dime and sometimes just spin in circles, but a very smooth circle, not a jerky, wobbly circle. There's a bigger one cruising around there. What's that guy? Might just be out of focus. Makes it look bigger. Yeah, that's another one out of focus. This is a good example. They're normally that fast, and this guy's quite slow. Well, you can kind of see the flagellum when I focus through it a little bit. Let's go back. Yeah, it's a real subtle point to see the flagellum. You gotta get just the right focus. Anyway, so that's this little guy. So I'm not sure what this is exactly. I don't think it's chlorella, but it's definitely a little green flagellate. Doing some nice photosynthesis while it's swimming. Okay, so let's back out again. Look for some bigger game here. A little cluster of cells for something. Let's do a digital zoom on that. See what that is. Interesting. Not sure what that is exactly. It's definitely a cluster of cells. It is greenish. So, some kind of plant-based thing, but not quite sure what that is. This is good pensive music. I'm not sure what that is. Do, 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 do. That's what I'm thinking about it. It's a mystery. Okay. It's another cluster of something. Looks like it broke up. So anyway, yeah, leave comments in the chat if you guys are watching or afterward in the stream on the comments. Hope you guys are enjoying Hendrix's music. This is second or third time I used this album. It's quite nice. Ooh, that's really cool. This guy's tumbling. That might be no, it's not a Volvox. It's not blobby enough. But it is cool because it is tumbling. Wish I could fix that frame rate thing. I have no idea why it's got a frame rate problem right now. Okay, let's do this. Let's go back out of digital zoom and do a proper eyepiece change or objective change and focus and try to get a better image that way. Yeah, it's not so cool that way. This objective is really not my favorite. It's kind of like the wrong numerical aperture setting for this scope. It's the same brand, but it has some issues. They're getting some really weird artifacts with it. Some like trails and stretching and stuff. It's really annoying. So I'm gonna have to go back to the other one and digital zoom it. That's the only way it's gonna stay nice and clear. Gotta replace that objective. Alright, where'd you go? There you are. So you can see it's tumbling away. It's kind of staying in place but tumbling a lot. And I don't know, again, the frame rate's bothering me. Camera's not in any special mode. Let me do this real quick. I'm gonna turn it off and turn it back on again. Troubleshooting 101. Reboot the device. It's still a little weird. And there's no setting in OBS that I tweaked. It's visually nice, but it's just a little choppy. It's supposed to be a little smoother than that. Still looks cool though. Just don't know exactly what this critter is. Other than a flagellated, it could be a colonial thing, it could be, yeah, sometimes colonial things will be in a ball of cells and each have individual flagella and they kind of tumble. That's why I thought at first by the movement it was like a Volvox, but Volvox are very green, usually very distinctly individual cells in like kind of a Death Star ball. And been looking for those for a long time. I haven't seen Volvox for years. I have to go buy some from a lab, Carolina Biological Supply or something like that. So we're buying some critters I can't find because they're really fun to study. So now it's taking a break and drifting. Going with the current, which means it may not be long for this world. Usually when the animals or plants stop moving like this, they got ready to lice and spew their contents all over the place. I don't even have the light on full blast, and I am using a bit of a diaphragm on it, so it's not full strength. So it's definitely not getting baked. Now it's at the margin of the cover slip, which makes it hard to look at. It's annoying. Come on. It's like an astronomical transit going through some shadow or structure and popping out. Like, oh look, the star's coming out again. Oh, there it is. Going. Now it's clear again, so you can see it. But now it's changing focus because it changed levels. It's coming closer to the eyepiece now, or to the objective. But it's back to tumbling again. So it's doing some kind of a swimming motion. Yeah, it's gone. I'll let him do his thing because that was a good enough view. I want to go look for a couple other things before I change slides. This slide is not as good as I had hoped. So let's just see what we can find. More cell clusters of something. Interesting. Zoom out a tad so I can do more browsing. Nothing really standing out. There's those little swimmers again, cruising around. Very fast. More swimmers, more debris. More swimmer. Lots of stuff that looks like it broke apart at some point. This one's more linear than the other ones, but it's similar to those other flagellates that were cruising around. Again, that frame rate's killing me. Don't know what's going on there. There's another frozen one that's stopped moving. Got some structure in there a little bit of something going on, but still pretty small. I'm zoomed in quite a bit, so I can't see that much detail. Okay, enough for that slide. That was kind of dull. A couple okay things there, but not too much to write home about. Let's try this one. Remember to wipe these cover slips off. Sometimes they come out a little bit dusty. Right out of the package. They're clean, but they're dusty. So let's try another sample from that same bit of water. So I'm going to stir it up a little bit. Maybe get something at a different layer. Whoa. It's already a cover slip on top. How did that happen? Okay. Let's go find the edge. There's the edge. Not too zoomed in. Let's go back out. Yeah. Got the covers up at an angle. Nothing. Those little swimmer dies. It's going really fast. Is it still jerky? A little jerky still. Interesting cluster of cells there. And some debris around it. It looks like something had broken. Let's zoom in a bit there, you can see. Unusual. I got some nice color there. Some greens. There's a little guy right next to it that's interesting, but it's much smaller. A ball of cells that's just hanging out there. Let's try a dark film on this and see what we get. There's oblique lighting. A little 3D pop. Interesting. And there's another thing up here. Let's check out a dark field again real quick and see. Oops, not quite dark all the way. Back to where you can see the colors a little bit, and then there's this other thing up here. It's also interesting. A little grouping of cells. Now we got some dark field there. Flagellate swimming around there, tiny little guy. And go back to oblique lighting a little bit. It's kind of fun, it gives almost a negative image at this level. Back to oblique lighting again. Kind of a mixture of oblique and dark field. So it's a tiny little dude swimming through there. Oh, it's just debris, it looks like. Alright, let's see what else we got at this. Right. Oh, there we go. There's a swimmer again. Going crazy. But it's damn choppy. Don't know why it's choppy. Very choppy. Got a big air pocket already expanding here. Oh, there's some filamentous algae. Oh, this guy's lysine. Look at that. It's like outgassing bubbles. That's wild. Breaking for some reason. But look at the trail it's leaving behind. That is crazy. And it's a little choppy, unfortunately, but it still looks cool. Look how long the stream is going. The current is carrying it way the hell down here. Let's zoom out a level. It's like a comet's tail shooting way down there. So something's causing it to eject its contents. Whatever it is, I don't even know what it is. But it's got a lot of contents. That's a trip. Oh, now the current's blowing the other way. That's wild. Hey Paul, how's it going? Native, yes. Sorry I missed you guys. I was too busy looking at this crazy shit that I found. It's bizarre. So there was a stream going one way, some kind of gradient causing it, and now it's going the other way again. Some kind of osmotic gradient. The slide hasn't been on that long, so it's not drying out yet. Oh, there we go. Now it's starting to go the other way. And I'm not breathing on it or anything. There's a certain kind of weird shift going on the slide. I'm just going to leave it there. I don't want to screw up the view. It's fun to see it shift back and forth. But at first it was a tiny little spout, like it sprung a leak. And it was just a nice little, almost like a particle simulator in 3D. And it was spewing, and then the current was blowing it nicely down to the left, down to the left corner, like a comet's tail. Now it's doing who knows what. Now it's static. Still outgassing, but no current. This is abnormal, whatever it is. Something has broken open. Normally, I don't even know what this is at this point. It looks... Can't even say what it is. It could be a clump of cells. It could be... It doesn't look like a typical single-celled critter. It's just a clump of something, but it's ruptured and spewing contents. Now you're getting some kind of a... there's no breeze in the room, and I'm not breathing on it. I'm about 2 feet away from the slide right now with my head, so I'm not sure why it's shifting. Sometimes if I breathe on it by accident, you do get these currents, and it makes a huge difference on the slide. But I'm just here enjoying the show. It's quite an animation. Now it's back to streaming that way. Now it's ramping up. Now it's back to the stream it was doing before, pretty much. You can see the same direction. Now I'm going to zoom in again because that was cool. Watching it kind of explode out of there. And you can see all the particles are dancing with Brownian in motion as the slide is warming up from the light. Now they're kind of scattering. Perfect with the music. It's got a little particle dance music. Whoa, look at that. And again, this, like I said, this slide wasn't on very long, so it didn't have time to dry out, didn't have time to get too hot. Whatever this is just decided to rupture. Let's crank down the light levels a little bit, you can see the particle details a little better. Whoa, now the current's going the other way, but it's still spewing particles. Wow, that is crazy. Again, I'm not breathing on the slide and there's no breeze in here. Some very strange water dynamics on the slide. Never saw this before. So bizarre. And you see, it's not even time to me talking. It's just got its own little rhythm going on. Oh, something goes swimming by, saying, "What's going on over here? Where's the particles coming from?" I mean, it could have been something that was like a tiny seed of something was stuck in the water, and eventually the water made it rupture. from osmotic pressure, because I've only had this water for about 24 hours right now. It's in the fridge most of the time. But it's a hell of a lava lamp kind of action. This is fun. This would be great for a background for some weird music performance, like a live performance put on the big screen. This is exactly why I haven't touched it yet. I want to keep this footage for something. These natural things are kind of hard to animate like this. This is so bizarre. Whoa, look at that. I think we just got a massive... I think the air bubble finally expanded really fast and just overtook the whole thing. Yep. Check that out. That's what it was. If I zoom out, you'll see it's already enveloped by the air bubble. And the water's to the right, so it's already in an air pocket. Those particles are no longer going to go anywhere. And now the particles on the outside are being chased. There's a whole bunch of them here. There's a cloud of particles over here. But if you go watch the edge of the air bubble, and the particles are being moved that way. This is the typical, you see the pinching and growing of the air bubble. There's a piece of debris there holding it back, so it's kind of growing around it. And it's going around that algae now. Slowly pinching off. I'm going to zoom in on this algae before I miss out on it. I haven't really studied it yet. I was too busy looking at that weird explosion. And here you can see all kinds of activity inside the algae. Look at that. The cells are alive and kicking. Look at all that activity in there. Let's do a little dark field real quick before we lose everything. Look at the cell activity, it's going bonkers. Let's do a little bit of oblique lighting real quick, just because we can. And you can see the compartments of the cells there very clearly. That's cool. Not sure which filamentous algae this is, but it's small enough to be, could actually be a, no, it's got the right shape and color to be an actual true algae, not a cyanobacterium. But it's not going to be here for long because that air bubble's coming. Start seeing that wall coming this way. I love seeing all the cell activity there. Look at it, it's like a party in there. Still a little choppy, but not horrible. Probably kind of the best view right there. I'll just leave it there because you get some color, you get some structure, a little bit of 3D pop. This is great about this high-res Nikon camera is I can, here comes the wavefront. I can digital zoom a lot because there's a lot of megapixels to play with, and have to change eyepieces less, so I have more flexibility. But it is nice having those contrast adjustment sliders on the microscope software, but it didn't seem to be compatible with this camera, so I had to use the Nikon webcam utility, which is putting all the control on the camera itself, so I have to keep messing with the camera directly and not the software. There's the air bubble. Alright, let's zoom back out and see what's going on here. There goes the air bubble. Oh, there's a dude up here. Look at that ciliate. I didn't even see it. Nice. Catch him before he gets fried. No, get away from that zone. You're going to get toast. I'm going to zoom back out on this guy. He's pretty cool. I like this guy. Again, lots of stuff going on in his little cell. Get a dark fill on real quick. Oh, and he got wiped out by the... Where'd he go? Oh, sent him flying. Didn't he get squashed? No, hope he didn't get squashed. So the algae's still doing its thing. Party going inside the algae. Where did that ciliate go? Wow, look at all this stuff over here. There's a lot going on here. That's cool. I think my ciliate friend got squashed. There's this little structure here too, which is odd. Not sure what's going on there exactly. And then there's this elbow junction here. Might be some kind of a reproductive structure there, I'm not sure. You got this weird thicknesses here. Off this guy. Oh, it's getting squashed on all sides. Alright, let's look ahead and see if we got anything. What's that? What the hell is that? Oh, there's a, now that is a cyanobacterial filament. Look at that. Undulating, very, very small. So it's like a colonial, small micro-bacterial colony called cyanobacterium. Nice, long, and wavy like that. They're connected as a colony, and they use flagella to move. There's a whole bunch of them here. When I change the exposure, you can see a bunch of them swimming around. That's cool. But you see how very small they are. I'm zoomed in a lot. And then there's another free swimming little rod character here. Another cyanobacterial thread moving on its own. That's a big colony. So they're all connected, but they're individual cells working together. There's a little colony. Nice scale there with the other one next to it. So the bigger one looks more like a typical algae colonial thread, whereas the one next to it cruising around and those tiny skinny ones are more of the cyanobacterium types. They're much thinner, much more agile, tend to be swimming around or moving around or flexing around a lot more than the typical algae does. Algae being a true plant, whereas cyanobacterium is related to bacteria because it doesn't have a nucleus. The genetic material is RNA floating around inside the cell matrix. So that's where the term prokaryote versus eukaryote comes from. Eukaryote means true kernel, kernel being nucleus, and prokaryotes are before the kernel. They don't have a nucleus, they're more primitive. But they still make pretty cool little structures like this guy cruising along here. They're doing just fine. They're still green, they're making their own food, but they're also swimming around looking for a better place to be. Whereas these larger algae threads just have a different approach. They just create these large filaments that collect into algal mats and just kind of hang. Some might dry, some might live, but they're just pretty much static. They don't move. But these guys move around, which is cool. That's a much better, well, it's a different survival strategy. Maybe it's not better, but it's definitely more fun to watch than some static filaments just sitting there hanging out. Although the detail inside these cells is quite interesting. You can see all the cellular machinery bouncing around. I'm not seeing cytoplasmic streaming with like chloroplasts going in a little circle. Sometimes you get that in some plants, which is really cool. To get the most light exposure and surface area covered, they stream their chloroplasts around, so they're constantly getting bombarded from all different angles of light. And there's probably other reasons for it too that I don't remember, but it is fun to watch. It's kind of a 3D look there. It's oblique lining. Pop a little bit. Very vermiform, worm-like, but not as shaky and wiggly as a nematode, where it just is constantly flexing longitudinal kind of muscle filaments back and forth, and this kind of frantic wiggle. This is more purposeful and directional. So although it's more primitive than a nematode on the evolutionary scale, look how very successful it is. It's moving around, it's making its own food. It's very, well, there's a nice color view there. It's doing quite well, being a prokaryote. Although it does have those little structures in there. Maybe this is one of those really flexible non-prokaryotic algae threads that can move around. But just based on the size and stuff, it's very much like a cyanobacterium. Ooh, we got some diatoms up here. Look at that. Some nice long glassy diatoms. Not much content in it, so they're pretty much dead. There's just a silica shell left behind, also called a test. In fact, there's a whole collection of diatoms here. Look at this. You go through this mess. There's the long spiky one. There's a more of a tapered one here. There's this little ring thing there. I don't what that is. A ring of cells there. This one up here is one of those tapered ones. Like a cigar. See those a lot. Cigar diatoms. And you got that classic yellowish brown pigment. It's still photosynthetic, but it's not your typical green you get in something like chlorella or some of the other filamentous algae or even just regular vascular plants. It's all kinds of stuff in this slurry here. All clustered. I kind of, when I got to the water collection, I also kind of stirred up the bottom of the shallow pond and then tried to collect some of this water. So this is probably where that came from. All the stuff that sank to the bottom that I stirred up. Yeah, all this activity around these algal filaments. It's almost like a long linear forest. You go hang out where the filaments are and you'll see lots of other creatures in the area. Getting this wavefront coming in, it's going to wreck everything, so let me just check along the wavefront. Looks like another diatom shell was right there. The deceased one. So nothing else along that wavefront. Check the rest of the slide down here and just see what I'm missing. Zoom out a little bit so it's not so crazy with the scrolling. There's the wavefront. Okay, so nothing there. Nothing down there. There's a wavefront in the other direction, so this is all being sandwiched. What's in the middle? Let's check the middle. Back up to the filament. Here. Back up here at the top. Let's see what's up top. There's the filament. Oh, sorry, there's the wavefront up there. There's a nice little building block of something that died and decomposed. Nature's brick wall there. What else can we find before it's too late? Looks like some other diatoms there potentially. Or something else. Some loose, big chunks of cells from something. There's one of those, no, that's something else. I don't know what that is. So yeah, I'm not getting much else on the slide. And it's just about dried out anyway, so time to swap. That's kind of fun. There's some good stuff on that one. That was nice. Alright, let's go to another one. Let's do one more from this guy because I'm just getting exhausted at this point. But I did want to take a little piece of the leaf and kind of scrape it. Let's see if there's anything on the leaf. So we can pull this out with the forceps. This is a piece of the lily pad that was in the water. So we'll kind of scrape it a little bit. And put that on the slide with some water. made a difference. Doing a scrape this time. A scrape and a dip. Scrape and a dip. What's those little tiny guys swimming around again? So nothing new there. Fix this lighting here. Some weird. Start with a little bit of a dark field. Zoom out a tad. So, a bunch of debris there. More little crazy wiggles. Oh, there's another cluster of those little things. That's they were bursting before. That's what I zoomed in on before that was breaking apart, whatever the hell it is. Kidney bean looking things. That's what was breaking apart before. Can't tell what's going on inside of them though. I don't know what they are. But if we're... okay, they may start breaking like the other ones were. Quite small actually. No, not super small, but small. Maybe the biggest one there is maybe half the size of a paramecium, something like that. Hey Grace, thanks for coming by. Got some choppiness on this camera for some reason today, but this is my Nikon mirrorless camera instead of the old USB cameras, so its image quality is much better, but for some reason it's choppy. You're listening to JX3D's music. I'm streaming here. His album called, I'm just forgetting the title. This is bad. Worlds Apart on his Bandcamp page. It's a very fitting stream for one of my astronomy streams as well as this stream, so I think it's the second or third time I played it. And so if you go back earlier in the stream on the replay, you'll see one of these erupting all kinds of particles on a wonderful stream, and then as the slide started to dry up, they created a kind of a flume effect, and the particles streamed away like a comet's tail. It's a very interesting visual. I still don't know what these are though. Never collected this water before. This is from a little pond feature near the biology department at Foothill College, where I was at my friend's radio show the other day, doing some wacky sounds on the air, and I got some samples while I was there. Nice air bubble. Air bubbles in space. Not much else going on this one. Lots of debris. Lots and lots of junk. There's an algae filament. But not much going on there. It's pretty dead. But I'll zoom in just for fun. a second I thought that was a vorticella that was squashed, but it is not. It's just some weird clusters of stuff. But there is definitely an algae filament there. Half of it's dead. It's pretty transparent. But there are some structures further in. Let's do a digital zoom on that. Let's see if we got any activity going on here. Some little flagellates bumping around, but not too much going on in the cells. There's some regular Brownian motion, but not like a lot of cell activity. Right, bacteria. Alright, well that slide kind of sucked. So, let's go with the good stuff that I got today from the zoo. I had a couple live streams earlier today on my synthetic channel from the zoo, where I got these water samples I'm showing next. Got some sloth footage because it was really choppy when one time I was on Kent's show. Frame rate was terrible. But today I got some really nice live stream footage of the sloth and some various birds doing all kinds of stuff, the raccoons. It's quite nice. So the first sample is from a little waterfall feature that runs down into the pond. It should be quite different than the stuff you find in the pond. The pond is usually the most amazing stuff. But let's see. Okay, let's go. Grab a thing here. See what's in this one. I should wipe this cover slip down. These damn cover slips, they come new, but they're all dusty. Must be like fine paper dust or something from the tissue paper inside. Alright, let's see what we got here. This one's usually fairly boring, but I wanted to try it again. And I got some plant fibers. It's kind of bizarre, but not that interesting. Oh, what's going on there? Something in the frame. Oh, it's the diaphragm thingy. What do we got here? Pretty sparse. Not a whole lot going on here. I gave up on trying to fix my frame rate thing tonight on the camera. I don't know why the camera's being a little choppy. But still got some good stuff. Just makes the fast swimmers look a little choppy. Okay, literally nothing on that one at all. Very boring. Water's very clean, as expected. Just wanted to verify that. So I'm going to give up on that sample because I've already been doing the other ones for so long. Let's go to sample number 2, which should be the best. 3 might be interesting too because it's in the raccoon area. And they tend to make a big mess in their water ball, as well as having some miscellaneous plant material in there. Might attract a bunch of critters of some kind, and also birds fly in there and bring in whatever they bring on their feet. So, But the pond's one of the best. I mean, my first best stream ever, and a few have passed it since, was from pond water I got about a year ago. It was stunning. I mean, I had so much stuff on one slide. I was just like, what the hell is this? It's a smorgasbord of critters. Some I've never seen before, some I haven't seen for a long time. So it was quite amazing. So I'm expecting good things every time I get samples from that pond. This is where the flamingos eat and crap, and the ducks are there. The peacock struts through there once in a while. There might be some critters coming in there, frozen food as well. So you never know until you get it all together. And then whatever, again, birds bring in from the outside. And stuff that comes in on the wind and all kinds of stuff. Alright, what do we got here? What do we got here? Too close, too close. There we go. Alright, let's see, let's see. I didn't grab stuff from the top or the bottom, kind of in the middle, so I'm gonna have to shake it up a bit. Little bits of stuff. Nothing interesting yet. Nothing interesting just yet. Okay. First slide was a total dud. But that happens. Let's try this one. I'm gonna shake it up a bit so it's got some spread on it. Oh, I also forgot in the same bag I have a feather and some rabbit fur to look at. I was shed by the rabbit there. I'll do that later. All kinds of stuff here. Alright, shook that up. Let's see what we got now. I may have had him collect too much water for me out of that section because it dilutes the sample. And some of the good stuff might be strewn about. Alright, what do we got? Okay, there's already a guy spinning out over here. What's going on right there? He's going bonkers. There's that frame rate problem again. It's annoying today. Let's do a dark field here so we can see what's going on with this dude. Definitely a flagellate with that crazy zigzaggy action. Going bonkers. But he's moving around too much to get much of a good tight shot of him. It's also going up and down through the water, so his focal, the depth of field is really shallow. But it's spinning. Spinning like it's at a Grateful Dead concert. Slow down, dude. There are tricks to slow these guys down. You can add thickeners to the water like methyl cellulose. It's the same stuff they use for slime in the movie Alien. And it's like they're swimming through peanut butter. Because they're already got a lot of, I mean, you think water is pretty mellow, but when you're this small, it's actually quite a bit of effort to swim through water already. So when you add something really thick to it, it slows them down a lot. And because it's clear, it doesn't mess up your view of them. You can see his little cilia moving stuff around there. Now, since it's not going too far, I might be able to push it one more bump. There we go. Spin, spin, spin. Spin, 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 little guy. I don't recognize this one, so I'm not sure what it is, but it's definitely a little flagellate. And it's not pigmented, so it's an animal, not a plant. It doesn't have any kind of greenish pigment to it. I don't see any little red eye spots or anything. It's very spinny though. You see tiny little flagellates in the background here and there, but also it's stirring up some particles when it's swimming as well. Spin, spin, spin. So this one's behaving quite well, actually. It's kind of doing its own thing in circles, which is great for close-up shots. Because when you have to chase them around, it's like the worst video game in the world. You gotta control 3 axes at once. And the Z, the focus axis, is the worst, because sometimes they put too much water in here. And you're just constantly focusing just to get an image at all. So it's not really fun. This guy's behaving pretty well though, staying mostly in the same focal plane. Just going back and forth. Let's try a little bit of oblique lighting on this guy, get a little 3D pop. No, don't do that. Stay here. There we go. Now you can see the organelles a little bit better because of the oblique lighting. It's got some definite structure inside there. What's fun is the large spherical things tend to have this concave look to them. It's like that experiment where you do the face with the different lighting conditions and the concave face suddenly looks like it's convex. Knocking that debris around. So that's a pretty cool one. Let's go look for some other fun characters where the slide starts to dry up. Oh, that's why I didn't even notice. It was in the area outside the slide cover slip. That's why I was swimming like crazy. Had a lot of latitude to go up and down because they're not as confined. Luckily, he was still spinning in a single focal plane and not going up and down. Alright, let's fix this so it's a little brighter. So we can find things better. Fingers crossed for some good stuff here. These little, ooh, look at that. It's a whole little cluster. Yeah, here we go. We got some good stuff here. So those little threads going in all different directions, those are cyanobacterial filaments, very, very thin relatives in colonial form of bacteria. And there are some little ciliates inside there. Let's go zoom in, see what's doing. This is a piece of their food. It's kind of a seafood chow with shrimpy bits and all kinds of stuff, but great food for the this little. Oh, there's a vorticella right at the end there. Look at that. That's a vorticella right there. Looks like a little wine glass upside down, but it doesn't seem to be filter feeding. I wonder if it's still alive. But the one on the left is just extended itself. That one on the left there is alive right there. And they're on these little stalks, and then often retract the stalks in kind of a coil. And it's really fun because it's like a defensive thing. And so there's a ciliate cruising around. There's a ciliate on stalks. These vorticella are ciliates on stalks. Let's get a little closer to the vorticella. See if it's going to filter feed. They filter feed like little, kind of how clams and mussels filter feed. Except instead of a hard shell, they just open up. There, you just retracted because it got disturbed by something. But you can see the little thread it's on. So it makes like a wine glass shape. And this is not the best exposure here. Let's see, can I make this a little more interesting? Digital zoom's giving a little bit of artifacts, but I didn't want to change the eyepiece right away, but maybe I should. Let's go back out. Go up and objective. That's much clearer. That's good. So now you can see the kind of a wine glass shape there. There's the triangular and a little stalk at the bottom. If I defocus it, you can see the stalk. And now I zoom in. Get a nice little view of it while it's trying to filter feed here. You can see the cilia at the top are kind of beating. Kind of have to change the lighting to see that better. Let's go back to oblique lighting a little bit. And now you can really see the vacuoles inside, those little spheres. And at the top, there's some wiggly motion. On the top, basically on the left side of the left margin, it's the top of the wine glass, basically at an angle. And it has a crown of cilia around there, funneling goodies into the opening there. And the opening has an interface there where I can create a food vacuole around a particle of food that it likes, and then brings that in and makes those little spheres that you see inside the cell. Those little, if I focus through it, you can see little spheres in there. Those are food vacuoles. Like microstomachs. Now you can really see the food vacuoles in those little spheres. Moving around, they got some yummies in there. And occasionally you can see the exocytosis where the food vacuole goes to the margin and they basically release it out into the environment, almost like defecating or vomiting, however you want to look at it. And the unused particles get pushed out into the environment. I've only captured that a few times on some of these cells. It's pretty fun to watch that. But you can see all that pulsating near the left side of the animal there. That's the crown of cilia fluctuating, making a vortex, hence the name vorticella. But I love the little squiggly wine glass. I don't know if my cursor shows up on this or not. You guys see a cursor here? Can you tell me if you see that on the picture and move it across? I can't tell if this camera software will show that. I changed camera software. check on my phone, see if my stream is showing that. Don't think you can see the cursor. I think it's only in the actual astronomy, the microscope software proper. You can see the cursor, but you can't see it in. Leave it here. I'm checking on my phone. Just got a delay. Yeah, it's not showing the cursor on here. Oh well. It's helpful though when I want to see the. I think OBS has a setting to override it and show cursor at all times, but I don't know where that is. Under settings, output, new, advanced, stream, general, information, HUD, cursor over projectors, make projectors always on top, no, overflow, multi-view, yeah, I don't see anything to show the damn cursor all the time. I'll play with that some other time. I don't want to waste stream time on that. Anyway, one of those little filaments on the right side of this little vorticella cell. You can see it kind of connected to the base there. It's a little kind of a squiggly thing, and it's helical. There you go. Now you just retracted on it, and you see it kind of coils like a spring, almost like a helix, and then it straightens out again. It's hard to tell in this lighting, but I have some much better videos of vorticella that show it. But this is a decent little view of this guy. Really see the contents of the cell. Let's go look at the other one down here. There's another vorticella down here attached to this debris. Right there. That's more the typical shape, like a little champagne flute or whatever. This one, yeah, this one's still pumping. I can see the cilia moving. This one has a lot of vacuoles in it. Look at all those little spheres inside there. This one's been busy. Chomping. But all these other filaments here are all cyanobacteria. Oh, it just retracted as well. Oh, there's the coil there at that lighting. You can see the coil really well. That's cool. He's got a very long stalk. There's the coil again. See it uncoil. I'm going to leave it right here so you can see the coil. It's nice. There's the coil. Look at that. That's so cool. It's like slinky. Boy, it's really getting disturbed by something. Come on, dude. Calm down. OBS properties of video source show cursor. Oh. Interesting. Thank you. Video source. Webcam. Properties. Custom audio device. Use hardware. Yeah, it doesn't have it for this app. Because I'm using an app and not a video source, it doesn't have the option for showing cursor, which is okay. I'll experiment with that some other time. But I love this vorticella wigging out here. This is pretty cool. Seeing that. Oh, thanks, Grace. Thanks for hanging out. Check out earlier. There was some pretty fun stuff earlier with things exuding particle streams and had a little botany lesson in the beginning about plant color, plant pigments, and fall color leaves and all that stuff. This guy's got some colorful food inside this one. See some pigments in there. Anyway, it's just as well I have the cursor turned off because sometimes I leave it on the screen too long and I go, oops, it just ruined my video. So it looks like, I'm not sure what the bigger ciliate is off to the right in here, the upper right. But it looks like we've got a, might have a little euplodes cruising around. Those are kind of fun to watch because they tumble and they got this kind of concavity to them. Oops. That didn't work too well. Didn't mean to zoom out. This guy's a really long stalk on it. I don't usually see him with stalks this long. It's pretty cool. But it's great when you see that coil. I think those cyanobacteria filaments bumping into it are bothering it, so it keeps retracting. There's all those filaments around it, probably disturbing it. So every time it retracts, those all shift around. And because he's touching some other ones, he's never fully retracting out. So it's kind of funny. He's getting caught up on those little filaments. That's really weird. I've never seen that before. Look at all those vacuoles in there. It's been a hungry little critter. Some of them even have the nice particles, the color from the debris above, which is very orangey, because the seafood in this food that the boy is really retracting a lot. Poor little guy's getting upset. See all that orange pigment in there? He's been eating little fragments of that. That's why he's got some orange inside of him there. But up here is a couple of ciliates here. There's this guy, I'm not sure what it is. Free swimming, it's not a Vorticella. And this little dude here looks like a euplodes. He's got his very, very distinct behavior of kind of cruising around, exploring, and kind of flipping around. I saw a couple of views looking earlier, and it looked like it was one of those little guys. It's super active. Uh-oh, here comes the wave front. Right there. It's coming in to do some damage. And there it goes. Doesn't take much. Just wiped out that little dude on the side here. He just got squashed, I think, in here. And so did that other little guy. But Vorticella's still on the bottom, so it's got a chance. There's two of them left here. And this guy's going to try and retract, and pretty soon it won't be able to go anywhere. Because that wavefront's going to come and ruin its day. See, it's trying desperately to pull, and it's getting denied because the wavefront is much stronger. That air bubble's pushing in big time. And now he's going to get sandwiched. Poor little guy. He might even get detached. Sometimes they get detached from their little filament and then become free-swimming Vorticella. They have to grow the filament again, I think. Oh, damn, it came from the other side. Ooh, this is dramatic. Let's go back to this view. Might be a little more interesting. So these poor guys are trapped with full food vacuoles they'll never get to enjoy. So they're still connected, but they can't go anywhere. The one on the upper side is a bigger version, and it's a little pinned right now. The other one has a little bit of room to move. There's still more water around it than the other one, but it's probably trying like hell to retract that filament, and it's not going anywhere. Yeah, the one above is getting too much pressure. See? It's starting to distend. It's going to lyse. So that huge vacuole forming inside the other one, it's going to pop. The smaller one's is okay, but the bigger one's not doing so well. I think it's been, because it got pinned, it's affecting the function. Its contractile vacuole is not pumping water anymore. It's going to take on too much water and pop. Because they're always fighting osmotic pressure from the outside, since they're sailing on the inside, just like we are. Certain amount of salt concentration. But that classic shape where it's all distended like that, and that one vacuole gets really, really big. I've seen that a lot on some of the other ciliates when they're about ready to pop. And there's not much else I can do about it, because as soon as I take them out of this slide, I pull the cover slip off. They kind of get destroyed anyway. They don't normally survive that. Unfortunately, it's the unfortunate reality of studying these things is they kind of sacrifice themselves for the good of the show and the good of science when people are doing research like this. But it hasn't popped yet. But it's definitely under a lot of pressure because that air bubble is coming in from all sides and it's getting smashed against the debris. Not a good place to be. See if I can tweak the view a little bit further here. Oh, come on. No, come on. So again, the guy at the bottom's okay, but see, he's also developed some of those large spaces in his cell. And it means he's not working well. Oh, the other one just lysed pretty much. You can see the debris is being contained, but it's already smashed. The other one is getting there. But the other one above is so fuzzy now that it's already popped its cell membrane in some area because it's getting really fuzzy. Let's go. Let's do it this way. See, it's kind of still there, but it's kind of fuzzy. No, maybe it is intact. Change the focus a little bit, you can see the membrane. But it's definitely not a happy camper. just had to change focal settings a little bit. But it's being restricted. They're both being restricted from moving, so the filaments are still connected, but they can't do anything about it. At least it's still surrounded by water. They're basically in a little pocket of water surrounded by air. All the stuff to the left and the right of that funnel shape is all air, no water. Or water droplets, basically, but not swimmable water. They're still keeping their shape, though. Usually by now they would not be contained. But there's no more movement, so probably any minute they're going to pop, so I'm going to spare them that, get them off the warm slide. Let's try another one. This is a pretty decent batch so far, but nothing extraordinary. Although he did tell me that he had just thoroughly cleaned out the filter basket there yesterday, and unfortunately that's where the good stuff accumulates. So if I had come yesterday, I probably would have got a lot better stuff out of this sample. It's usually teeming with goodies. But it's always worth checking no matter what. And then I'll check that raccoon water and see how that looks. Oh, too many covers close. So get that out of there. Okay, what do we got on this one? Hey, there we go. We got a ciliate. Yeah, this is a fast one too. Holy moly. Alright, let's fix that exposure. Check the zoom. There we go. Get that stupid thingy out of there. Okay, zoom in a little bit. And let's see what we can find. Okay. Nothing there. Lots of colorful debris from the food from those guys. Flamingos have very colorful chow. Okay, some little ciliates here. Doing their thing. Little milky looking little ciliates. Let's zoom in a tad. Why is that looking so funky? Whoa. No, not like that. Alright, let's go back to that setting. Turn the brightness down there. Okay. There's that big guy went flying through earlier. This little guy is doing some stuff here. Oh, why is it doing that? Go back. Oh, that's weird. Why is that alignment off there? That's really weird. Okay, so there's that little... Wow, how's that going offset like that? There we go. Wow, it's really... How does that get so far offset? Oh, I must have toggled this guy. I've got my zoom center off. Well, it's way off. So over this way? No. Is it down? Or up? There we go. That was funky. I was totally losing track of stuff because I bumped the zoom center. So where were we? Little guy down here. Very nice. Let's just fix the exposure there a little bit. And this a little bit. Still choppy a bit. I gotta fix the camera setting somehow. But this is a nice little specimen. Little cilia going through debris. And a little flagella off in the distance to the left. Wiggling like crazy. It's coming in out of frame. It's over here. So let's look at the debris field. It's not too dense. I don't have to focus up and down too much. Pretty good. It's kind of one of those nondescript little roundish oblong ciliates. So I'm not sure what this one is, but it's another one for my list to figure out. On my little research. There aren't very many good field guides on this kind of stuff if you guys haven't looked, so you kind of have to cross-check a lot of visual websites, and some are quite easy to figure out, some are much harder. Kind of like if you ever try to look up a specific species of spider. There's very few good spider books out there. I mean, you're lucky if you get it to like family of spiders or something like that. Like an orb weaver or something like that, but which is basically not even a family. I think it's an order. I think it's Arani, which is way far down. So that's like we're order Primates, and so family's Hominidae for hominids. So if you go, oh yeah, I got the order, well, that's not really that far. I mean, for some things that's perfectly fine. Like a lot of insect orders, that's pretty good. Like the dragonflies and the damselflies, and the beetles, and some other stuff by their orders. That's perfectly decent. At least it's a beetle or a dragonfly or a thrip or something like that. But certain things you definitely want to go further down than order if you can. But for some of these critters, it's just not a, if you're in the industry and academia, or you're studying this stuff for other reasons like water quality or something like that, and you study them all the time, you'll find lots of good references probably in those places, and they have a good online database that's a private one. But online, it's really hard to figure some of this stuff out, especially to species level. Like the fact that Amoeba proteus is so prevalent means it's pretty easy to identify, and yeah, it's Amoeba proteus. That's genus and species like Homo sapiens, and that's a pretty easy one. Or Paramecium caudatum, which is one of the most common paramecium species out there. It's most likely what you're going to find in a lot of places. But some of the stuff, like these Vorticellas, I don't know how many of the species there are, but I know most of them is Vorticella. I don't know what the species is. But to get to that far down is pretty good in a ciliate. Unless it's like an extremely common one. So anyway, not much on this slide to be honest. So let's make another one. And again, because the trap was cleaned recently, it might be why I don't get as much good stuff this time from that pond trap, which is perfectly okay. I always go back, but it's a great place to start because I almost always get really good stuff out of it. So can't run it every time. Let's do one more from the pond trap, flamingo pond trap, and then we'll do some from the raccoons. See what we get from the raccoons. No. I didn't do the fish tank they have there this time, the cichlid tanks from Lake Malawi in Africa, because it was incredibly boring last time I did it. I got like literally nothing out of the water. So the water is very clean and very recycled through this filtering system and stuff. So there was nothing to report at all. Oh, really nice long filament. Look at that. This is a huge filament. Let me zoom way out. Look how long that is across the whole field of view. Massive algal filament. That's crazy. All the way down here. All the way off the slide. One giant filament. That's crazy. That's the longest one I've ever found. And it's bending and curving, so it's not even a straight line. It's going all the way across the entire cover slip. Yep, almost gets to the other side. But it takes a lot of turns and twists. So it's probably like twice the length of a cover slip. This thing's huge. Let's zoom in and get a good look at it. Because of its thin nature, I'm assuming it's cyanobacterium. They're often quite thin and long. Let's do a little dark field action, turn the brightness down, and then zoom in a tad more. And get a little focus. So we're getting some green color, which, there are plenty of cyanobacteria
filaments are greenish. Oscillatory is a famous one, and it's a pretty chunky one. But this one I don't know what it is. It's very nondescript. There's nothing like real distinct about it. But just the fact that it's so damn long is crazy. A little more oblique lighting there, give it a nice 3D look. So it's incredibly thin, which again leads me to believe it's a cyanobacterium thread. Most algae just don't get this thin. And it's incredibly long, it's crazy. Let's go back to kind of normal view here to get some color. A little more dark field for the contrast. I'll zoom out a tad. Really appreciation of how damn big this thing is. It goes on and on and on. Goes down, goes across, off the cover slip, and wiggles out to there. Crazy. Not seeing much else on the slide so far other than this, so let's take a look. It's a pretty darn boring slide. So thank goodness for that filament. There's a couple little tiny ciliates poking around the background. But that filament, yikes. That's nuts. Am I zoomed out all the way? Looks like I am. Try and snap a quick picture of that if it'll let me. Looks like it's working. Zoom in, do a little bit of a adjustment again. Let's try and snapshot that a couple times. It's really hard to get an idea of the color at this. I have to zoom in a lot to get the color. That's right about where I get the color. That right there. Zoom in on those later. Okay, well, this time the pond catch filter wasn't quite as good as it should have been. That's okay. It happens. Let's check out the raccoon. And about that time, I'm getting ready to call it quits. It's been a long stream. Going on for, lost track how long it's been going. It says only it's about 2 hours, but... Oh, looks like the stream crashed. Policy violation. Huh? the fuck happened there? Policy violation. It's my policy violation. What did I do this time? Well, that's unfortunate. Stop